Hey everyone, this is an exam question about therapeutic cloning, and this is taken from the Combined Science Biology Paper 1. This is from 2018 and this is from a higher paper. So this is relevant to everyone. And this question is particularly useful because we have this really clear diagram here, which I'm going to talk us through to help you understand the process. Figure 10 shows how embryonic stem cells are produced in therapeutic cloning for use in patients. So therapeutic cloning is a way to produce embryonic stem cells, and embryonic stem cells are useful because they can help to repair damaged organs and possibly in the future cure diseases like diabetes and things like paralysis as well. So we're aiming to get these stem cells from an embryo because these embryonic stem cells, remember, can differentiate into any new type of cell. So the process involves the patient, so the person that needs the treatment, a normal cell, we call that a somatic cell. So body cell, if you hear the term somatic, it means a body cell is taken for the patient, so for example a skin cell, and the nucleus is removed. So this thing here, this black dot, is the nucleus. That will contain all the genes for that patient, and it will make sure that the embryonic stem cells have the same genetic makeup as the patient. That nucleus is transferred into an empty egg cell from a donor. So quite usually this donated egg cell comes from a different person, not the patient, and that could be for a few reasons. It could be that perhaps the patient is male, so doesn't have egg cells. It could be the wrong age or have health issues, which means that their egg cells can't be used. An egg cell comes from a donated person, a donor. The patient's nucleus is transferred into it. This essentially creates what would be a fertilized egg. It's sort of like a zygote, essentially. So a fertilized egg with a full set of chromosomes, and then it will be treated in the lab with some sort of stimulus to help it divide and produce an embryo. So after a few days, these cells are harvested, they're removed from the embryo and they're placed in the culture, and then they can be treated to produce things like pancreatic cells, for example, that would produce insulin for someone with diabetes. But it's very important to note that the growth of the embryo is halted after a few days. So it doesn't develop into an embryo with working organs or anything like that. It's halted before that happens. So now we need to look at the advantages and the disadvantages of therapeutic cloning in medical treatments. We need to think of two of each. So let's start with the advantages. So this type of treatment can be used to cure current diseases, but it can be also potentially used to create diseases in the future as the technology develops. So the curing of medical conditions is the main advantage. It's also important that you use the nucleus from the body cell of the patient, because this means that these cells won't be rejected by the patient. So the patient won't mount an immune response against these stem cells. So the cells are unlikely to be rejected by the patient. So other advantages that you could mention are that you can produce cells or tissues of any type. Many cells can be produced. You can also go on to use these cells for research, not just for treatment, but for scientific research. And it also allows waiting times for organ transplants to be reduced, because these can potentially be used to build a whole new organ. Any of those could be used. So then let's look at the disadvantages. So there's kind of ethical arguments which make up the disadvantages. The main one being that the potential life that this embryo could be, were it allowed to grow on to eventually become a fetus and then a baby, that life is potentially is destroyed, is prevented from happening. So that would be the main disadvantage. There are some risks of egg donation or egg collection. There is also a shortage of people willing to donate their eggs for this procedure. It might be that this has some side effects on the person donating the egg cell. There's a few potential other disadvantages. Let's go with the risk of egg donation. And some other disadvantages are that this can allow tumours to form, this process. it also has quite a poor success rate in actually producing this viable embryo in the first place. But these two for each are plenty to give you 4 marks. I hope that helps.